So, welcome back to the lecture series once again. So, we are starting the fifth week. So, last week we talked about in depth about the AFM setup and how AFM setup is coupled with electrochemistry setup and how these emerging integrated tools are changing the face of biosensing. In the last class, I highlighted upon one point that modern biosensing research is much of a multimodal deal. That means multiple techniques of detection are coupled in one single device. So, in one device you will have microscopy, you will have antibody labeling, you will have electrochemistry, you will have techniques like AFM, you will have techniques like for surface plasmon resonance, you will have techniques like phage display. So, instead of doing or performing these operations in individual units, now the trend is changing, and the reason for change in trend of having multimodal devices or multimodal setup is the amount of analyte, the amount of sample is very limited. So, when you have very limited sample, you cannot afford to waste sample. So, that is why, because of the extremely low titer, much of these technologies are now very integrated multimodal technologies, and whenever you are pulling together multiple techniques in one platform, you are always have a issue of extreme noise, because every modality brings its own set of noises and own set of signal to noise ratio into the system. Apart from it, in order to understand the systems, you have to have a complete understanding of multiple techniques in one platform. So, more and more this whole field is a huge design endeavor of individuals with varied expertise and technical know-how. So, with this small brief up, I will start this fifth week from lecture 21 to 25. So, coming back to this circular map, what I have been showing from the beginning, you see that what we are detecting is in the center. It could be in different form, it could be vegetative form, spores form, they could go dormant or they could be active, and there are features on them. They could have toxins, DNA, surface proteins, DPA and others. And then on the outer periphery, you have different ways of detecting them. So, lateral flow immunoassay, which you will be studying this week, nanosensors, electrochemical sensors, optical sensors, mass and Raman spectroscopy, DNA sequencing, DNA protein chips. Now, when we break up this diagram, it is something like this. You have these targets. These are the targets what you have to detect. So, the vegetative cells or the spores or the fragment of DNA or the toxins or the surface proteins or DNA. So, this is that second circle what we are talking about. Now, in terms of the biosensor configuration, there are these recognition elements and signal generation devices. So, it is the recognition element which are being recognized and they generate signal, and the recognition elements are monoclonal antibodies, single chain antibodies, peptides, aptamers and phage display techniques. Now, while proceeding with some of these techniques like SCM, and of course, I will be proceeding with SPR, LFIA, then eventually PCR on a chip, nanomaterials, I touched upon some of these recognition elements, but I haven't touched upon in-depth understanding of this recognition element. So, this week, a part of it will be devoting about how these different recognition elements are generated. Generation of recognition element and their mass production and the technologies involved in their production. So, in terms of the recognition element, these are monoclonal antibodies, what we will be discussing, the first one. As the name indicates, it's mono means single, clonal means it's a clonally produced, monoclonal antibodies. For those of you who are not from biology background or coming from other backgrounds where they haven't heard this word, so first word what you have to understand is antibodies. What are antibodies? So whenever there is a foreign element which enter inside your body has a capability to distinguish between its own and what is not its own. So when there is a foreign element which is not part of the body, it generates an array of proteins which tries to capture this foreign material. So it's something like, so there is a foreign element, say X, which is inserted into the body. So what this individual will do, in order to counter X, it will produce from its own body something called minus X. So this minus X is going to nullify this X. So this minus X, which is generated by your own body, is called antibody. And this X, which is injected or got into your body, is called antigen. And this whole orchestra of antigen antibody reaction is the whole field of immunology. Now, when we are talking about any pathogen entering your body, be it a virus, be it a bacteria, be it a foreign DNA, be it a spores, be it a fungus, be it a foreign protein, body will generate antibodies against all of them. And these antibodies are very, very specific. And some of the ways this antibody works, so say for example, this asterisk shows your foreign material. So what will happen is antibodies will come close to them and they will surround them like this. So this Y kind of thing what I am drawing is the antibody AB, and they will nullify these antigens and will clear up your body. So when the body fights, it is basically against some pathogen. It is the antibodies in your body which are fighting against the antigen. So in order to prevent you from disease, for any kind of diseases starting from cancer to bioterrorist agent, body will be producing antibodies. And these antibodies, now if you look at the charts, these are those recognition elements, because corresponding to a spore, corresponding to a vegetative cell of pathogen, corresponding to a DNA, corresponding to a toxin, corresponding to a surface protein or a DPA, body produces extremely specific set of molecules or antibodies. So, if you know the antibodies, what you can do is you take a blood sample and if you know the antibodies which are being generated against it, you will be able to detect whether this pathogen is present in the sample or not. This is how this whole thing works. And in order to detect, you have all these different signal generation devices, including quartz crystal microbalance, surface plasmon resonance, LFIA, PCR on a chip, nanomaterials, and of course, the signals could be in the form of fluorescent signature, color, DNA amplification, electrochemical signature, SPR angle, piezoelectric, Raman and EIMS, etc., and all these other techniques. So, you understand it is a very integrated approach. So, if you do not know about the antibodies, it will be very challenging for you to understand the whole concept out here, what we are going to talk about. Now, what are monoclonal antibodies? Now, next thing. So, in terms of definition, we talk about what are monoclonal antibodies, also called MO, that is monoclonals, and ABS is the antibodies, or MABs in short. These are the short forms, are proteins made in laboratories that act like proteins called antibodies in our bodies. Antibodies are part of your immune system, as I mentioned earlier. They seek out the antigens or the foreign materials and stick to them in order to destroy them. In terms of sticking to them is this reaction what I was showing you. So, for example, these are the antigens, then the antibodies will surround them like this. They will locate them in your body and they flow in the blood vessels. So, most of these antibodies travel through the blood vessel and the lymph vessel and kind of run after the foreign material and go and capture it and neutralize it. This is how it works. So, coming to monoclonal antibodies, they seek out the antigens and stick to them in order to destroy them. Laboratory made monoclonal antibodies help stimulate your own immune system. The word monoclonal refers to the fact that the antibodies created in the laboratory are clones. As I mentioned, these are all clones. They are exact copies of one antibody. The generic name of the product often include the letter MAB or monoclonal antibodies at the end of the name. And monoclonal antibody is a very, very powerful tool in immunology research as well as in biowarfare and bioterrorism research as well as in biosensor research. Now, you will also come across a word called polyclonal. If you come across this word mono, then you will come poly. Poly means many. Okay. What is the difference between monoclonal antibodies and polyclonal antibodies? The difference between the two types of antibodies is in the names. Mono refers to one and poly refers to many. Monoclonal antibodies are clones of just one antibody and they bind to one antigen only. Polyclonal antibodies, on the other hand, comes from several different types of immune cells and will bind to more than one antigen. So, as the definition says, it must be clear to you that monoclonal antibodies are far more specific versus polyclonal antibodies. So, whenever people does immunocytochemistry or immunohistochemistry assays, if they find a strong binding with one monoclonal antibodies, that gives them more authentic confirmation that the reaction is working in the right sense. So, this is the first difference between monoclonal and polyclonal antibodies. How are monoclonal antibodies used? Monoclonal antibodies are used for diagnosis, disease treatment, and research. Now, when we talk about diagnosis, this is the area of what we talk about in the field of biosensing. And when you talk about the disease treatment, it is essentially is when this monoclonal antibody is there to neutralize the foreign or the disease element. So, monoclonal antibody therapy is a promising therapy with its own pros and cons, but definitely it find application in diseases like cancer, neurodegenerative diseases, and in a huge domain of biosensing as probes to identify material in laboratories or for use in home testing kits like those of the pregnancy or ovulation. So, most of the pregnancy kits use monoclonal antibodies. This is the second popular application. To type tissue and blood for use of transplants. This is basically, most of you have seen blood testing, where you are testing the blood or testing the tissue. So, these kind of blood testing and tissue testing for transplantation. So, you want to graft a particular tissue from one person to another person. Most of this screening is being done with monoclonal antibodies for diagnosis and for disease treatment. Now, in terms of monoclonal antibodies have been treated following conditions, as I mentioned, it is used for against cancer, cancer cells. So, say for example, cancer cells are producing some specific antigen on the surface. So, if you could generate specific monoclonal antibodies against this specific antigen of cancer cell, then you can, if these are the cancer cells with the specific antigen on their surface, and if you have the specific monoclonal antibodies, these monoclonal antibodies will go and will surround these cells and will try to neutralize their effect. They are using for organ transplant rejection identification. They are used for inflammatory and autoimmune disorder including allergies, infections including COVID-19. There are monoclonal antibodies produced against COVID-19. Osteoporosis, a bone related disorder where there is degeneration of the bones. Eye conditions, migraines, high cholesterol level, nervous system disorders. So, if you see almost every spectrum of our health is being tackled at some point or other using monoclonal antibodies. So, it is a very, very potent and potential tool for wide array of disease condition, wide array of degenerative conditions in your body. And if you look at the number of US Food and Drug Administration, the approval of monoclonal antibody therapies has been rising since first monoclonal antibody which was approved in the year 1986. So, if you look at it from 1986 when the first time it was approved, so the technology was running much prior to that, almost 20, 30 years prior to that. So, 1986 to 96, 2006, 2016, and now we are almost to 2024. So, 10, 20, 30 and almost now 4 decades. So, monoclonal antibody technology is now ushering into almost the 4th decade or eventually half a century to travel, and a lot of breakthroughs, promises, and at times there are dejection, rejection, so many things. But Still, this is one of the most potent recognition element for biosensor research, and we'll study a little bit more how they are being produced in the next class about hybridoma techniques, about selection techniques, about phage display technique, and how they are produced in the industry. Thank you.